Hello and welcome to the Chapter 9, Part 3 lecture on the reactions of aerobic respiration, also known as cellular respiration. You should use the information in this lecture to complete the Chapter 9, Part 3 guided notes, which of course you should complete before you attend class. In Part 2 of this chapter, we started discussing glucose metabolism, or glucose breakdown. Remember that the point of that process is to break glucose down through a series of steps, pull energy out of that glucose molecule, and use it to make ATP. Remember that glucose is like a $100 bill, whereas ATP molecules are like $1 bills. So ATP molecules are easier to spend one at a time than a $100 bill. Now, in the last lecture, we took glucose through glycolysis and broke it down into two pyruvate molecules. And at that point, I told you that there's several different ways we can go with our pyruvates. In part 2, we took it over here into fermentation, and we discussed how glycolysis paired with fermentation is a very effective method of making ATP if you're a small cell with low energy needs. Fermentation supports the process of glycolysis and allows those cells to continue doing glycolysis over and over and over again. Glycolysis makes 2 ATPs at a time from a single glucose molecule. So if 2 ATPs at a time is enough for you, that's fine. However, what about cells like ours? We have animal cells and they have high energy needs. We're very energetic cells. 2 ATPs at a time is not going to be enough. So what can we do? Well, this time we're going to take our two pyruvates over here into the mitochondrion and take it through cellular respiration. Notice a couple of things about this reaction. First, it requires oxygen, O2. It is aerobic. This is the reason that we breathe oxygen, is to power this process. And the second is that it takes place inside the mitochondrion. So we're going to have to go in there to get this process started. Let's take a look at it. The endosymbiotic theory tells us that mitochondrion are essentially little bacterial cells living inside our own cells, and they help provide our cells with energy. There's some parts of the mitochondrion that you need to be familiar with before we get into the reactions of cellular respiration. Like all cells, mitochondrion have an outer membrane. This is just a phospholipid bilayer like we find on the outside of our own cells. Mitochondria are sort of unique in that they have an inner membrane as well. They have a second layer in here. And this inside layer of phospholipids is folded into these little wrinkles that we call cristae. Now, between the two layers is a space. So there's the outer, there's the inner, and you'll notice there's a little gap in between filled with fluid. We call that the inner membrane space, and some important reactions are going to occur there. The last part of the mitochondrion that you need to know about is called the matrix. It's this liquid in the very center inside the inner membrane. So that's the matrix. So regarding cellular respiration, where are we going? Well, we have our two little pyruvate molecules, and what we need to do now is to take them all the way inside the mitochondrion into the matrix, so into that liquid in the center. Now notice that in order to do that, we have to go through two membranes, the outer and the inner. Now because pyruvate is kind of large, it's going to need to be actively transported into the mitochondrion twice to get into that matrix. That's actually going to end up costing us a little bit of ATP at the end of the process. When we count up how much we spent and how much we made, this step costs us a little bit. But that's okay, it's worth it. You're going to see we're going to make a lot of ATP. So let's head into the matrix and begin those reactions. The first reaction of cellular respiration is known as pyruvate oxidation. Sometimes it also goes by acetyl-CoA formation. Either name works. As the name implies, in this reaction, pyruvate is going to get oxidized or broken down. So it has 3 carbons in its backbone, but the product at the end is only going to have 2 carbons in its backbone. So this molecule got broken down. The remaining carbons that are left over get added to another molecule, and it forms this thing called acetyl-CoA. Also during this reaction, we have some CO2, carbon dioxide, that's released, and we release enough energy from that exergonic reaction to make an NADH. Let's take a closer look at it. Let's look at pyruvate oxidation in a little more detail. Here's good old pyruvate. Notice that it has 1, 2, 3 carbons in its backbone. Once the pyruvate enters the mitochondrion, it's going to go through a step wherein the top part, this top carbon with its oxygens attached, is going to get removed by an enzyme. That little molecule is going to form a molecule of CO2, which is going to leave. Now think about it. Every time we breathe out CO2 or carbon dioxide, right? Well, this is one of the sources of that carbon dioxide. Now the remaining product, that remaining 2 little carbon chain, is going to go inside the mitochondrion and it's going to encounter this thing. This thing is called coenzyme A. It is a multi-enzyme complex, so it's a big complicated molecule. And the job of coenzyme A is really to grab that little leftover piece and carry it into the next step. So it's like a little transport molecule. That's all it does, is deliver this to the next step. Here you can see that coenzyme A has grabbed on to the 2 carbon part that's left over. Sometimes a 2 carbon chain such as this is referred to as an acetyl group. So collectively we call this molecule acetyl-CoA, and that's what's going to go into our next step. Now remember what happens whenever we break a big chain like this. When we take a 3 carbon chain and snap one of the carbons off into a 2 carbon chain, energy is released. That is an exergonic or catabolic reaction. So those energized electrons that are snapped off during this process are going to be numbed up by NAD+, and it's going to end up forming NADH. So the NADH becomes reduced and is energized, and we're going to use it later on. Notice that no ATP is made during pyruvate oxidation. There's one other thing you should notice about pyruvate oxidation, and that has to do with the numbers of molecules we're dealing with. This diagram shows a single pyruvate undergoing oxidation and forming a single acetyl-CoA. But remember that from our original glucose molecule, we actually had 2 pyruvate molecules to work with. So if we have 2 pyruvates to start out with, we're going to end up with 2 acetyl-CoAs total. So this reaction will repeat twice. We're also going to end up harvesting 2 NADHs from this process. And it requires 2 coenzyme A's, and it makes 2 CO2s. So remember that total through this reaction, because it runs twice, we have to double the numbers of all of our molecules, if we're keeping track of them, which we are.
As this mountain biker heads up the trail, the breakfast he ate this morning is being burned to power his bike ride. His breathing rate increases as his leg muscles demand more oxygen to burn more fuel. Let's zoom down to where this fuel is burned, our cells. Here, the blood vessel on the left delivers fuel and oxygen to a single muscle cell. In cellular respiration, energy in fuel is converted to ATP, shown here as starbursts. Most ATP is made in the cell's mitochondria. ATP powers the work of the cell, such as contraction. Let's take a closer look at how ATP is produced from a molecule of glucose, our fuel. Only the carbon skeleton is shown to keep things simple. The first step is called glycolysis, and it takes place outside the mitochondria. To begin the process, some energy has to be invested. Next, the molecule is split in half. Now, the molecule NAD+, an electron carrier, picks up electrons and hydrogen atoms from the carbon molecule, becoming NADH. Keep track of the electron carriers. They play an important role by transporting electrons to reactions in the mitochondria. In the final steps of glycolysis, some ATP is produced, but not much. For every glucose molecule, only 2 net ATPs are produced outside the mitochondrion. However, glycolysis has produced pyruvic acid, which still has a lot of energy available. Let's follow this pyruvic acid molecule into a mitochondrion to see where most of the energy is extracted. As the molecule enters the mitochondrion, one carbon is removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA. Coenzyme A is removed, and the remaining 2-carbon skeleton is attached to an existing 4-carbon molecule that serves as the starting point for the citric acid cycle. The new 6-carbon chain is partially broken down, releasing carbon dioxide. Several electrons are captured by electron carriers, and more carbon dioxide is released. The carbon dioxide that you exhale comes from the reactions of cellular respiration. 2 ATPs are produced by the citric acid cycle for each molecule of glucose. At this point, only a small number of ATPs have been produced. However, more energy is available in the electrons that are being transported by electron carriers. While the citric acid cycle starts another round, let's follow an electron carrier to the next step in the process. Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondrion. The chain consists of a series of electron carriers, most of which are proteins that exist in large complexes. Electrons are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy. The oxygen you breathe pulls electrons from the transport chain, and water is formed as a byproduct. The energy released by electrons is used to pump hydrogen ions, the blue balls, across the inner membrane of the mitochondrion, creating an area of high hydrogen ion concentration. Hydrogen ions flow back across the membrane through a turbine. Much like water through a dam, the flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in your cells produce most of the ATP that is generated from the food you eat. The process you've just observed, cellular respiration, generates 10 million ATPs per second in just one cell. That ATP can power a biker up the trail, or it can power your brain cells as you learn challenging biology topics.
The next major step of cellular respiration is the most complicated. It's a big giant cycle known as the Krebs cycle, or sometimes called the citric acid cycle. This cycle actually consists of 8 different steps catalyzed by 8 different enzymes, but we're going to keep it pretty simple. Essentially what happens in this cycle is that this molecule from the last step, acetyl-CoA, is going to go in, it's going to be affected by a number of enzymes, and a number of different energy products are going to be harvested and made, including NADH, FADH2, and ATP. This is the last step of glucose breakdown, so this is the last point at which we can get any more energy out of that original glucose molecule. A waste product of the Krebs cycle is CO2, which again we breathe out. Let's take a closer look at the Krebs cycle. The Krebs cycle can be broken down into 3 phases. Let's introduce each one. First, the acetyl-CoA from the last step is going to join up with a molecule known as oxalacetate, and a molecule known as citrate will be formed. In the second phase of the Krebs cycle, the citrate is going to be broken down through a series of steps, and energy products will be formed as a result. In the third phase of the citric acid cycle, that 4-carbon intermediate that we end up with is going to get recycled back into oxalacetate so it can start the cycle over again. Surprisingly, this recycling step will actually release a couple of energy products for us as well. Let's take a closer look at each phase. Here's the first phase of the Krebs cycle. There's 2 major reactants. oxalacetate, and the acetyl-CoA molecule that was formed in the last step. Here's our little acetyl-CoA molecule. Now the coenzyme A molecule that delivered it here is going to drop it off, and then that guy's going to leave and go back to the previous step to pick up more acetyl groups and deliver them to the Krebs cycle. So he's going to leave. That leaves us with the acetyl group, which is a 2-carbon molecule. It has 2 carbons in its backbone. Now here's oxalacetate, which is kind of fun to say. Notice that it has 4 carbons in its backbone. When the acetyl-CoA and the oxalacetate get together, they end up forming this molecule, citrate. And if you look at the molecule closely, you'll see that it has 6 carbons in its backbone. Now that makes sense because 4 carbons plus 2 carbons equals a 6-carbon molecule. Citrate is also known as citric acid. So that's the reason for the other name for this cycle. It can be called the Krebs cycle after the scientist that characterized it, or it can be called the citric acid cycle, because citrate is citric acid. In phase 2 of the Krebs cycle, citrate from the last step is going to get broken down a couple of different times so that energy can be harvested out of it. In this first step of the second phase, citrate, which is a 6-carbon molecule, is going to get broken down into an intermediate that only has 5 carbons. So in other words, a carbon was snapped off of the backbone. Now where does it go? Well, it leaves as CO2. That's a waste product that we breathe out. Now because that reaction is exergonic, because we broke something, energy is going to be released, and guess who's going to capture it? Good old NADH. This reaction is going to happen again. This intermediate, and don't worry about learning its name, it's going to start with 5 carbons in its backbone. A carbon's going to be removed, so we're going to end up with another intermediate with only 4 carbons. Again, because a carbon was removed from the chain, that carbon is going to leave a CO2, which is waste, and energy is going to be released, which can be snatched up by NADH. In the last phase of the Krebs cycle, that intermediate that we were left with, which incidentally is a 4-carbon molecule, is going to get recycled back into oxalacetate. Now oxalacetate is also a 4-carbon molecule, so no carbons are going to be added to the chain, but stuff is going to get rearranged around that backbone through a series of steps. Now it might surprise you to learn that these steps are actually exergonic. These are catabolic reactions that happen through this rearrangement. So energy is going to be released. Now who's going to grab up our energy for us? Well, there is an enzyme present that is capable of substrate level phosphorylation. In other words, it's going to take an ADP and change it into ATP for us. So we make an ATP, that's great. Other enzymes are also present that will make an FADH2, becomes reduced and is filled with energy, and at another point an NADH is going to get filled up with energy as well. At this point, oxalacetate has been recycled, so that cycle can start over again, and it will repeat over and over and over again as long as the cell continues to need ATP. That complicated Krebs cycle was a lot of work, so what do we get out of it? Well, first let's look for each acetyl-CoA. One acetyl-CoA enters the cycle at a time, and from that, the following are made. 2 CO2s that are breathed out as waste, 1 little ATP, and incidentally, this ATP is made by substrate level phosphorylation. That means that a little enzyme took an ADP and a phosphate and squished them together, if you remember. We also managed to make 3 NADH molecules and a single FADH2 molecule. Now remember that this cycle actually has to run twice in order to harvest the maximum amount of energy from the molecules we have at hand. And we had 2 acetyl-CoAs, so it'll run twice. That means you have to double the numbers. From the 2 acetyl-CoAs that we got from breaking down glucose into pyruvate and pyruvate into acetyl-CoA, we actually end up with 4 CO2s, 2 nice ATPs, 6 NADHs, and 2 FADH2s. So this process is useful because you harvest a lot of these energy carriers and you actually get to make a little ATP, which is the point of this process. The Krebs cycle actually marks the end of the glucose breakdown part of this metabolic pathway. So at this point, we've broken glucose down as far as we can go, and we've harvested as much energy out of it as we can. Before we move on to the next step, let's assess where we are. First of all, let's look at ATPs. How many ATPs did we make through all these steps? Well, we only made 4. We made 2 profit in glycolysis and 2 in the citric acid cycle. So that's pretty good, but it's not nearly enough to power an energetic cell such as an animal cell. So what else do we have to work with? Well, let's look at our energy carriers. NADH, we managed to make 10 of those guys. We made 2 in glycolysis, 2 more in pyruvate oxidation, and 6 more in the citric acid cycle, so 10 total. We also managed to make a little FADH2. We only made 2 of them from the citric acid cycle, but they're going to be handy too. Now, NADH contains enough energy to make 2.5 approximately ATPs. So we can make quite a bit of ATP out of the energy in these NADHs. There's also enough energy inside an FADH2 molecule to make 1.5 ATP molecules. So we can use that energy to make some more ATP. Well, how do we do that? For that, we're going to need a machine. We're going to feed these energy carriers into a big machine known as the electron transport chain. And that chain is going to convert that energy from the carriers into ATP energy. Let's take a look at it.